Good morning. My name is Lindsay Holden, and I am a PhD candidate at Portland State University in Portland, Oregon. I am also a SMART scholar based at the Toxicology Directorate at Army Public Health Center on Aberdeen Proving Ground in Maryland. So every day, we are all exposed to small doses of toxic chemicals. The air we breathe, the food we eat, the water that we drink, all has little tiny bits of bad stuff in it. But for the most part, everyone in this room, we're all fine, healthy human beings. So the big question that my research aims to answer is, why is it that some people, these green people in this diagram, why do they get sick while other people don't get sick when they're exposed to the same chemicals? There are three main factors that go into this. The first is your exposure. So that is how long you're being exposed to a compound, so the length of exposure or time, the concentration of the compound that you're exposed to, so the dose, and then the route of your exposure. Are you breathing it in? Are you eating it? Are you putting lotion on your skin? Another aspect is the basic demographics of who are you. There are physical characteristics, like are you a male or a female? Are you young or old? Do you have a high body mass index or a low body mass index? Additionally, there are behavioral characteristics. Do you work in a job where you're exposed to weird chemicals more than average? Or do you smoke cigarettes and are you exposed through chemicals in that mechanism? Additionally, there are health characteristics such as asthma or diabetes that might make you more disposed to being sensitive to a certain suite of chemicals. And then the third aspect, the part that I'm interested in, is the genetic component of whether or not you're susceptible to chemical exposure. So there's this interesting study that just looked at a single compound. In this case, it's BAP is the chemical that they looked at. And this beautiful, colorful map of the world is showing the concentrations of this chemical across the world. So blue means there's not a lot of this chemical there. Red means there's a lot. But the concentration of that chemical doesn't always correlate with the risk of getting some disease associated with exposure to that chemical. So in this case, if we look at Russia, Russia here has an increased prevalence of this disease. It's a 1.5% prevalence, but Russia has a lot of blue in this chemical map, whereas Southeast Asia and India have a lot of red, but their exposure risk is a lot smaller. So concentration of a specific chemical doesn't always mean that you're going to be at the super high risk. So maybe there's something interesting about each of those discrete populations of people, that something about their genetic makeup makes them more or less susceptible to some chemical. So specifically, I'm interested in one type of difference at the genomic level, and that is copy number variation. So let me tell you what that is. Copy number variation is when we have large deletions or large duplications of our DNA. Usually we have one copy we get from mom, one copy we get from dad, so 2 copies of every gene. But sometimes you can have a deletion of one of those copies, or you can have duplication where you have 3, 4, or 5 copies of the same gene. And surprisingly, 12% of all of our genomes, and everyone here, 12% of your DNA has these large deletions and large duplications. Which when I heard that, it blew my mind. I said, how am I alive? I have all of these huge errors in my DNA. And so what does that really mean? Let's look at an example. So salivary amylase is an enzyme in your saliva. It's the first step in how we digest food. So in populations of us humans, they found that you can have anywhere from 2 copies, the normal copy number of this gene, all the way up to 15 copies of this gene. And if you take DNA and you stretch it out really thin, and dye it with a fluorescent tag, you can count the number of copies of DNA that you have. So in this bottom individual, they had 1, 2 copies. This person had 1, 2, 3, 4, 5, 6 copies, and then the person at the top had 14 copies of that gene. And what that looks like in real life is that you have more copies of the gene, so you have more of the protein being expressed. So that means individuals, in this case, that have more of this gene being expressed and they're making more salivary amylase, means they digest starch really rapidly in their mouth at first stage of digestion versus other populations. So Interestingly, copy number variation, it has some things that are conserved across all humans. So in this room, we all share certain large deletions or large duplications, statistically, mathematically speaking. But for each discrete suite of different populations across the globe, there are unique sets of copy number variants. So that would be all of these little colored areas. So there are certain aspects of these large deletions and large duplications that are unique to different human populations. So maybe that's a factor that's playing into this, what's causing different traits in different populations. And lucky for me, a researcher who uses zebrafish strains also have a similar copy number variant, specific suite of copy number variants to each strain. So let me tell you what I mean when I say strain. So human populations, you can imagine, okay, there's the, traditional Han Chinese population, or maybe there is a Yoruban population, so there's these very discrete populations of humans. In zebrafish, we have different strains. So one of the very common strains that's used, and I'll be telling you about 3 strains that I use in my research, is this strain AB. So the AB strain was founded in the 1970s. It was the original strain of zebrafish when this model started being used, where scientists went to pet shop A and pet shop B, bought some fish, bred them together, kept them going until the 90s. So that's strain AB. Then in the 90s, they did this really wacky technique called gynogenesis, which is essentially where they're taking a single parent offspring and building a population from that. So we've got this interesting population, AB. We also have Tübingen population founded in Germany, where scientists went to multiple suppliers and bought very distinct populations of zebrafish, bred them all together with the goal of making a very diverse population of fish. And then the third strain is a wick population, which is where scientists went out into the natural habitat of zebrafish in India, collected individuals, brought them back to the lab, brought them together, and that's the foundation of the strain that we have today. And we know a few things about these strains. We know that they differ at the genotypic level, so on the genes or DNA level. They differ at the single base pair level, and they also differ in the copy number variation that are unique to each strain, and that they differ at the phenotype level or the trait level. They all look generally the same, but they might have a different clutch size, so the number of eggs that they lay. They have different schooling behaviors, and interestingly, they also have different sensitivities to chemicals in these different strains. And so I'm interested in verifying that the differences that we see on the DNA level in zebrafish strains and some of those different traits that we see in zebrafish, that there's some sort of translation between DNA and phenotype or trait. And so by looking at gene expression, I found that yes, there are lots of differences in zebrafish. So yesterday we heard a little bit about different effects of stress in the different sexes. We learned about that in mice. And I found something very similar in zebrafish, which is not super surprising. Males and females tend to be different in many levels. But more interestingly is that the strains, so here each color corresponds to a different strain, are more different than differences between sexes. And if I drill into this is a lot of data compressed down into 2 dimensions. If I drill into that and I say, what are the genes that are different, I can make really confusing looking heat maps where dark red, so the more saturated a red color is, we can think of that as the gene being turned off. So it has a decreased expression relative to the other strains. And a dark blue color, more saturated blue, means that gene has been turned on. So we separate out males and females because we know they're different, and we look across all the different strains. We can see that each one of these columns represents a single gene, and there's about 450 genes here that are unique. Some of these overlap, but most don't. And you can pull out some interesting trends that, for example, this suite of genes here are all relatively turned off in the AB strain relative to the other 2 strains. Or we can look at this big suite of genes that are all turned on in one strain relative to another. So there are unique expression differences across these strains. And if you summarize that numerically, we can just see that there's lots of genes that are either being turned on or turned off in both males and females across all of these strains. So, what did I just say? We know that some traits are dictated by copy number variants. Cool. We know that the genomes of humans and zebrafish both have copy number variant regions that are specific to those populations in humans or strains in zebrafish, and that copy number variant regions can directly impact a trait, the salivary amylase example we talked about. And then, more importantly to me and my interests, is that both human and zebrafish populations have different sensitivities to chemicals. So, as I finish up my dissertation in the next year, I will be answering the big question of whether or not the genes that are associated with sensitivity to chemicals, do they cross over these regions that have large deletions or large duplications, and is that what's causing one strain of zebrafish to be more sensitive or less sensitive to a chemical, and can we translate that to the big picture of why some people get sick versus why others don't. So with that, I'll thank my mentor and my funding, and thank you.